Hey everybody, so talking immunology today. We're going to start, this is my first lecture, it's going to be on history, so let's just get into it. The basics of immunology is intrinsically tied to the history of infectious disease, and so we need to kind of establish the groundwork for infectious disease. Now I'm going to assume that maybe you've taken a microbiology course or had some kind of exposure, hopefully not literally, to an infectious disease, so you understand the basics, right? There's viruses and bacteria. And they can make you sick, right? Usually it was smallpox or measles. That was probably the biggest one. So smallpox is probably responsible for hundreds of millions of deaths, at least in the post BC era, so in AD. As far as going back even further, like into the BCs, early like 10,000 BC, even back that far, we knew we had infectious diseases because we have at least oral records from back then. And so we know that we've been dealing with these diseases forever. As long as humans have, there's been at least one human next to another human, there could potentially be infectious diseases. And so we've been dealing with those a lot. And so smallpox is the big one that's going to come up a lot. Measles is also a big one. So like one of the first written accounts that we have is from Galen. And they knew there was some kind of plague. We don't know if it was smallpox or measles. It's hard to tell sometimes because uh, now we know that those like pustules that they both form would be different. But I mean, back in the day, like I don't know if they just said they had pustules, so we don't know exactly what it was. It was probably smallpox though, because smallpox was the worst and killed so many people. It's absolutely crazy. And we've been dealing with it forever. And why am I talking about this? Well, because smallpox, yellow fever, measles. We're going to keep going. HIV, influenza, Ebola, and now we're on to coronavirus. All of these viruses and a bunch of bacteria along the way too, but a lot of the viruses were the stimulus for us understanding the immune system. And so, if we understand these infectious diseases and how humans are dealing with these infectious diseases, that's going to be kind of what gives us some clues into how the immune system actually works. And so, let's keep on going. The history of immunology really starts with, I'm going to butcher this, Thucyd- Thucydides. There we go. That sounds Greek. Thucydides, and so he was writing things down. That's him. They probably looked exactly like that. Look at that dude. He's got a beard and everything. He was in Athens, 430 BC ish, and he was writing down some ideas, and he wrote down that a plague, which it wasn't plague like as in the Black Plague, it was probably smallpox. It could have been measles too. But he recognized, and when I say he, keep in mind these Greek philosophers, there were a bunch of people doing this. Was it actually him that physically wrote it down? We don't know, but we're going to give him credit. Thucydides. But in one of the translations, it says, the same man was never attacked twice. So he's referring to the ability of people to not become reinfected. And that is the key. If you take nothing else away from this, the idea of immune memory is one of the, if not the key points of immunology. How do you actually defend yourself? Well, yeah, you have your innate immune system. That's like your barriers, like your skin that just keeps bad guys out. You've got macrophages that eat bad guys, and you got neutrophils that blow up and kill bad guys. And those are your frontline defenders. But to actually remember what a bad guy is, by bad guy I mean pathogen, that's really, really key. And as far back as 430 BC, we were able to recognize that, which is really, really interesting. Now, as we keep on progressing, now keep in mind I'm skipping over a lot of things, but we do know that around 10th century-ish, there were some monks, and I'm going to show a video in a little bit that talks a little bit more about this. There was a monk who took the smallpox pustules. So you can see here's a boy in, it looks like a 60s or 70s that's probably suffering from, well, it's not probably, he has smallpox. And so you can see there's all these pustules, and they'll leave severe scarring. It's really, really bad. And what they were doing was they would take either the pus or the pustules themselves, and they would dry them, and then they would blow it into the nose, i.e. an intranasal administration, which is something we still do, or an intramuscular inoculation. and we called that variolation. Now, that is different from vaccination, and I'll talk about that in a little bit. But basically what they're doing is they're taking weakened virus because it's kind of drying out. It's in this pustule that's kind of dried out and stuff, and they would physically dry it out as well. And then you blow that into the nose, and then, maybe 3% of people would still get the disease and die, but that's better than 30% of people dying, which is what was typical for the mortality rate. And that was smallpox. Smallpox was really, really bad. We are super, super blessed, I guess, to live in a world that smallpox was eradicated from. Smallpox was really bad. My parents' generation, so that's the baby boomer generation, they were the last generation-ish to actually be vaccinated because we wiped it out in the 70s, which is really, really interesting. And so I will show a video of that in a minute. Now, variolation, this idea of taking pustules or dried pus, sounds super gross, and actually giving it to people either through their nose or through like a little scratch on their arm. That variolation, that continued from 10th century China all the way up through the 1700s, up through like the mid to late 1700s, which is crazy. Now, here's something really interesting though. So in the 1700s, there was some folklore that was going around, and I call it medical folklore for lack of a better term. And they knew that people who had cowpox, which was just kind of a milder infection, you still got sick, you still got a fever, you still had these pox on you that looked kind of like this, but not as severe. They knew that people recognized that if you had suffered from that, that you were able to take care of the people that had smallpox and not get it. Well, that's really interesting. So your body is remembering an infectious disease, right? So immune memory, that's really, really cool. And so a guy who does not get enough credit was Benjamin Jesty. And so in 1774, he was an English dairy farmer. And so late 1700s, there would be these smallpox epidemics that would travel through, okay? And so they lived in a small little town, and they knew, hey, it's creeping up the coast. He's in England, and it's creeping up the coast or whatever, and it hit the town right south of us. It's about to hit us. You'd think people would be more careful, but they didn't know. Like, it was just an airborne disease. Like, it's really tricky. I mean, think about how coronavirus has spread. People, we know more about it now, and we still can't stop those spreads. So how are these people supposed to deal with it? They didn't have face masks. And so he said, all right, you know what? I've had cowpox before. He was a farmer. And he recognized that one of his milkmaids or one of his helper people had also had cowpox. And so he found a lady who had fresh cowpox, took the pustules, and kind of tested out this hypothesis that this folklore said that if you had had cowpox, you wouldn't get smallpox. And so he treated his wife and his two sons. And the account that I read was that people, his neighbors, thought he was absolutely crazy and like evil for doing this. But guess who didn't die from smallpox? His wife and two sons. Now, cut to Edward Jenner, who we generally consider the father of immunology, though that's a little arguable. He was much more scientific about it. So Benjamin Jesty, along with actually a couple other people, whose names are lost to the annals of history. But they kind of established the idea that it was, hey, maybe there's something to this. But Jenner really was scientific about it, and he conducted a bunch of vaccination experiments showing this correlation between giving someone cowpox versus smallpox. And he also coined the term vaccine. So I took 4 years of Latin in high school. This is like the only time I ever used it. Vaca, which that would be more of a Spanish-speaking version of it. Waka would be like the Latin way to say it. Is Latin for cow. Hence vaccine, because it was cowpox. Hence waka. And so I want to cut to a TED Ed video. Oh man, I love TED Ed videos. They do a really good job. And it's going to talk about the history of smallpox.
10,000 years ago, a deadly virus arose in northeastern Africa. The virus spread through the air, attacking the skin cells, bone marrow, spleen, and lymph nodes of its victims. The unlucky infected developed fevers, vomiting, and rashes. 30% of infected people died during the second week of infection. Survivors bore scars and scabs for the rest of their lives. Smallpox had arrived. In 1350 BC, the first smallpox epidemics hit during the Egypt-Hittite War. Egyptian prisoners spread smallpox to the Hittites, which killed their king and devastated his civilization. Insidiously, smallpox made its way around the world via Egyptian merchants, then through the Arab world with the Crusades, and all the way to the Americas with the Spanish and Portuguese conquests. Since then, it has killed billions of people, with an estimated 300 to 500 million people killed in the 20th century alone. But smallpox is not unbeatable. In fact, the fall of smallpox started long before modern medicine. It began all the way back in 1022 AD. According to a small book called The Correct Treatment of Smallpox, a Buddhist nun living in a famous mountain named Ome Shan in the southern province of Sichuan would grind up smallpox scabs and blow the powder into nostrils of healthy people. She did this after noticing that those who managed to survive smallpox never got it again, and her odd treatment worked. The procedure, called variolation, slowly evolved, and by the 1700s, doctors were taking material from sores and putting them into healthy people through 4 or 5 scratches on the arm. This worked pretty well, as inoculated people would not get reinfected, but it wasn't foolproof. Up to 3% of people would still die after being exposed to the pus. It wasn't until English physician Edward Jenner noticed something interesting about dairymaids that we got our modern solution. At age 13, while Jenner was apprenticed to a country surgeon and apothecary in Sodbury, near Bristol, he heard a dairymaid say, "I shall never have smallpox, for I have had cowpox. I shall never have an ugly pockmarked face." Cowpox is a skin disease that resembles smallpox and infects cows. Later on, as a physician, he realized that she was right. Women who got cowpox didn't develop the deadly smallpox. Smallpox and cowpox viruses are from the same family, but when a virus infects an unfamiliar host, in this case cowpox infecting a human, it is less virulent. So Jenner decided to test whether the cowpox virus could be used to protect against smallpox. In May 1796, Jenner found a young dairymaid, Sarah Nelms, who had fresh cowpox lesions on her hand and arm, caught from the udders of a cow named Blossom. Using matter from her pustules, he inoculated James Phipps, the 8-year-old son of his gardener. After a few days of fever and discomfort, the boy seemed to recover. 2 months later, Jenner inoculated the boy again, this time with matter from a fresh smallpox lesion. No disease developed, and Jenner concluded that protection was complete. His plan had worked. Jenner later used the cowpox virus in several other people and challenged them repeatedly with smallpox, proving that they were immune to the disease. With this procedure, Jenner invented the smallpox vaccination. Unlike variolation, which used actual smallpox virus to try to protect people, vaccination used the far less dangerous cowpox virus. The medical establishment, cautious then as now, deliberated at length over his findings before accepting them. But eventually, vaccination was gradually accepted, and variolation became prohibited in England in 1840. After large vaccination campaigns throughout the 19th and 20th centuries, the World Health Organization certified smallpox's eradication in 1979. Jenner is forever remembered as the father of immunology. But let's not forget the dairymaid Sarah Nelms, Blossom the cow, and James Phipps, all heroes in this great adventure of vaccination, who helped eradicate smallpox.
You gotta love those TED Ed videos. They're so good. Okay, so cutting ahead, just to briefly talk for 1 second, because I'm in this history lecture, I'm trying to kind of discuss a little bit of the groundwork of some of these deeper concepts that we're going to encounter as we dig deeper into what's actually going on with immunology. And so this first example of a vaccine is a really, really interesting idea that touches on virology, microbiology, immunology, vaccinology, all these ologies. The idea of viral antigens or any kind of antigen and its relatedness, okay? And that's the key to cross-protective immunity. And so what we actually encountered, it was kind of just this fluke chance that this related virus happened to have its antigens match up close enough to not cause severe disease in humans, but to cause enough disease to initiate an immune response that was cross-protective. That's what we would call that. And so cowpox viruses are actually somewhat unrelated to the variola major and minor viruses. There are lots of pox viruses. There's horse pox, camel pox, all kinds of stuff. So you can see this is a relatedness tree. And so you basically take all of the genomes for these viruses and you throw them into a computer and you say, what are the odds that, how closely related are these things? And so you give it, out of 100 chances, how closely related is this? And so like 100% of the time, these split out this way. 100% of the time, these split out this way. When you start digging a little deeper, sometimes Okay, 69% of the time these are different from these, but so that means what? 21% of the time they're not. Um, so anyway, so it's just relatedness. And so you can see if we look at, this is orthopox viruses, so it's a family of viruses, which viruses are kind of goofy. They don't follow the traditional kingdom, phylum, family, order, class, genus, species. They kind of start with families, but it's all right. Viruses are just really weird. Anyway, so we can see that cowpox is related distantly, at least, so with a common ancestor here, to the smallpox viruses. The variola major being the one that usually hurt people the most. Now, the actual vaccines that we used were even more distantly related. Most of them came out of the South American cluster here. This is the most closely related. Um, this is a vaccinia virus. So all of these colored ones here are vaccinia viruses, and they're even more distantly related. But their antigens match up close enough to the smallpox or to the variola virus, which causes smallpox, to cause cross-protective immunity. And so that's where we were actually seeing it. Now, you may know if you get vaccinated against smallpox, or if you know, like maybe like your grandparent or something, they'll have a scar because the vaccinia virus would still cause at least a mild infection because it was like a live virus. We have different versions now, but we eradicated smallpox so people don't get vaccinated against smallpox anymore. Anyway, so moving on, cross-protective immunity. So we're going to keep on going, and so with the success of the smallpox vaccine, that led to a drastic increase in the number of people actually paying attention to fighting disease. Well, so keep in mind the time here. So time moved slower back in the day. And so Pasteur, who everybody's heard of, Louis Pasteur, pasteurization, he did a ton of stuff. Most of these scientists that we're actually going to talk about, these older scientists, oh man, they were all super prolific because there was so much space in the world to like study. Nobody knew anything, and so they all they all did like hundreds of different things. Whereas now, like you're super specific, like you know how to do this one tiny thing in science. Well, these guys did everything. They dabbled in all kinds of stuff. Um, but he was working on a vaccine against chicken cholera, which it sounds kind of goofy. They were trying to vaccinate chickens, but I have a chicken here with chicken cholera. It's still a problem. It's a Pasteurella bacteria. Okay, so not a virus, but a bacteria. Now, what a virus was versus what a bacteria was, that was still being developed at this time as well. And so it wasn't always super clear how to actually spread an infection, like what it was. They could see the bacteria, but if they were trying to study things like rabies, they knew that it wasn't bacterial, so they weren't really sure what was going on, but they did know that it caused an infection. So they could study that. And so his first vaccine that he developed was for chicken cholera. And he extended that concept of a bacterial vaccine into his work for anthrax, which was actually isolated by Robert Koch, he's on the next slide. So you know, it was all kind of happening in the same world. Okay. And so he developed, because anthrax was a big deal for herd animals, not just humans, but it was worse for herd animals because they could spread it. And so he was developing these bacterial vaccines, okay? The chicken cholera one was really interesting in that it came about by accident. So he told his assistant, hey, he's growing cholera, which is this Pasteurella, hence Pasteur, because, you know, he sort of worked on it. And so he said, hey, undergrad, go put the stuff in the fridge. I'm exaggerating on what really happened. And the guy was like, okay, but he misunderstood and he just like left it sitting out for a month while Pasteur, I guess, went on vacation. I mean, back then they could just, you know, he's like, I'm going to go to a conference, so I'll be gone for 2 months or whatever. So he came back and it had been sitting out and basically had died. And so he was like, well, let's try it anyway. And so he injected it into the chicken. And the problem that he had been running into was that when he would take his Pasteurella and inject it into his chickens, they still got sick. And he just, I don't know, he was having a lot of trouble developing a vaccine that didn't also kill the patients, or the host, we would say, until he figured out, oh, this one's dead, and we gave it to the chicken, and lo and behold, the chicken is resistant to reinfection. Awesome. He took that and into anthrax. It was working. Awesome. And then he made international headlines because he was working on a rabies vaccine. And now Pasteur, he was a smart dude, and he figured out the idea of passaging. Okay, and so without fully understanding that it was a virus, he still was able to develop a vaccine. And basically, what he did was something that we still do. and you take your pathogen, which he just knew that the saliva from a rabies infected person or dog usually was infectious. And so he took the saliva and he would put it on cells. Okay. And so what he was actually doing, he knew that rabies was, it seemed to be infecting, it would affect your brain, right? So because you start acting crazy and doing crazy stuff. And so he took the neurons from a rabbit's spinal cords and put those in a petri dish and put the rabies virus on there, or what he didn't know was the virus at the time. He put the saliva on there and it would grow, and then he would take the liquid from that, put it on some more spinal cords, more spinal cords, more spinal cords. He kept doing that. That's called passaging. You're passing it back and between different cells in the hopes that it will slowly become attenuated. Attenuation just means it's kind of like a weakened form of the virus. Like if you just took a rabies virus out of a dog, it really likes infecting dog cells, right? But if I take it out and I kind of force it to infect a rabbit, it gets used to living inside of a rabbit cell. And so I do that, 100 times, and then I try and put it back into a dog, and it doesn't kill the dog anymore. And so that's the idea of passaging. And he saved an 8-year-old boy who had been, I think the term was ravaged by dogs, which was terrifying. But he saved his life. They knew the kid had rabies. Or they were like, how could this kid not have rabies? And so they let him treat him with this vaccine, and it saved his life, which a good thing, because that's why we get to still have vaccines. Now, I mentioned Robert Koch a second ago. Oh, bitter rival. These two did not like each other. I don't have a good reason why. Just, I don't know, they just didn't like each other for whatever reason. There's people that don't like me. Maybe it's you, I hope not. But Koch was also doing a lot of simultaneous work. Now, Koch is arguably the father of modern microbiology as opposed to immunology, because his focus wasn't necessarily on immunology, though I did mention, all of these dudes, which it was mostly dudes, of course, all of these dudes were working on all kinds of random stuff. But he was really focused on purifying bacterial culture. He worked a lot on culture media and perfecting that so they could grow different kinds of bacteria, which then you could use to make a vaccine. So he was always kind of dabbling in that world. Koch's postulates, which you've probably heard of, the idea of establishing what is causing a disease. Now, of course, there are diseases that are exceptions to Koch's postulates. Postulates? But for the most part, you can use that to study infectious diseases or establish what is actually an infectious disease. But he did note, he's, I guess, mildly famous for noting some important aspects of acquired immunity, which we're going to talk about that later. That's like immune memory, basically. That's another term for immune memory. He noticed that in people who had malaria. So he went to Papua New Guinea and noticed that they could find malaria parasites, so Plasmodium, it was probably Plasmodium falciparum, but there's a couple Plasmodiums, sorry. They could find the Plasmodium parasite in the blood of people from Papua New Guinea, but they weren't really that sick, as opposed to when they had, I think it was Chinese workers or people from England would go to Papua New Guinea and get malaria. They got really, really sick. And so he noticed, he was like, how come these people, everybody's infected, but only some people are really sick, and these people aren't as sick, because it's kind of always been around. And so that was a really important idea that's going to be extended on in just a minute. And so he did get the Nobel Prize in 1905. He worked on tuberculosis a ton, including, he isolated tuberculin, which would just be basically the antigen. So if you ever got a tuberculosis test, they put a little tuberculin in there, and then if you react to it, after a couple days, then that means you've been exposed to tuberculosis and you need to go on antibiotics. Well, him and a few other people figured out like how to isolate tuberculin, but they thought it would be a really good vaccine. And the problem was he didn't, dot his I's and cross his Q's. What? Whatever a good analogy would be. And he was like, oh my gosh, we made a tuberculosis vaccine using tuberculin. It's awesome. And everybody was like, oh, that's great. And then they started using it, and it wasn't. We still don't have a tuberculosis vaccine that really works very well. And so it was a huge scandal. But they still gave him the Nobel Prize in 1905, so he didn't fail. He's probably most famous for his postulates, as well as anthrax. So he basically isolated and established what anthrax was, which then allowed Pasteur to make a vaccine against it. So good job, Bob. Moving on. So von Baren and Shibasaburo, I have to start saying it, Shibasaburo. So these two dudes were working under Koch's tutelage. They worked in his lab. Okay? And so he was working, and they were working in his lab. They're basically like research scientists, a little beyond postdocs because they were already established scientists in their worlds. A lot of them were MDs as well. The delineation between a scientist and an MD was only really established later on. Um, so like I have a PhD, I'm a scientist, I'm not a physician. But most of these guys dabbled in all of those. A lot of them started their careers actually. I think it was Koch that actually started his career doing as surgeons, and then they moved on. But they were very scientific about what they were doing, and they were working in his lab. And in around 1890, they developed what they called antitoxins. I called it serum antitoxins. We now would call this passive antibody administration, which is something that we do. Against diphtheria. That was the big one. And so they were both nominated for a Nobel Prize, the first Nobel Prize in medicine they were nominated for in 1901. They didn't give it to Shibasaburo, um, ostensibly because he's Japanese, I guess, because they're just racist. I mean, he was nominated, though. I don't understand why they didn't give it to him. It was mean. But von Baren got it. Anyway, so that was 1901. But they got that for their diphtheria antitoxin. So what they would do, they would take horses and guinea pigs and animals like that, and they would inject diphtheria, the bacteria, which is Corynebacterium diphtheriae. They isolated the actual toxin, and so this is really important. So some bacteria are so dangerous, not because they're growing inside of you, but because of the toxin that they secrete. The two worst ones being diphtheria and tetanus. So it's not the diphtheria bacteria that kills you, and it's not the tetanus bacteria that kills you, which is Clostridium tetani and Corynebacterium diphtheriae. It's the toxin that they secrete. And so you need to deal with that toxin. It's going to kill you way before the bacteria was ever going to kill you. And so you need to take the toxin and you put it into like a horse, and you pull the blood out of the horse, you isolate the serum, and you can inject that into a person, and that can keep them alive long enough for them to fight off the actual bacteria that's making the toxin. So it's sort of, it's an antitoxin. It's an antibody that inhibits the toxin itself. This is now, so we skipped over the idea of using the passive antibody administration, and now we just give people toxoid vaccines. So instead of injecting into a horse and taking the horse's serum out and injecting that into you, which we can do that in some cases if rabies is one good example for that. So if you get bit by a rabid dog and your rabies vaccine isn't up to, if you've never been vaccinated against rabies, then what they'll do is they'll start your vaccine regimen to start really boosting your adaptive immunity, which we'll talk about later in this course. But they'll also give you some passive antibody serum, which it's usually, I think it's human, but it's just a bunch of antibodies in like PBS, which is the like salt water. It's like what they put in your blood thing, the IV. Wow, that's terrible. So they would just put an IV in you, and then they give you a bunch of antibodies, and it's to block the toxin until you can make your own antibodies. And so, but toxoid vaccines are what we use right now. Really, really nice. So if you've ever gotten like your Tdap vaccine, that's what two of those are. The T stands for tetanus, and the D in the DAP stands for diphtheria. Pretty cool. Now we're cruising through the late 1800s. We're heading into the 1900s, and we're going to move on to one of the most prolific biologists slash microbiologists slash immunologists slash chemists on the planet, Paul Ehrlich. This dude, you got to go to his Wikipedia page and see all the random stuff that he's done. I mean, I remember he developed like all the stains that we used when I was learning immunology in grad school. Like, this guy just did so much stuff, and he had a friendly rivalry with Ilya Mechnikov. And so there were kind of two sides of the immunology coin. And so Ehrlich was a humoral immunity guy, and he thought that the humors, which is kind of that Greek carryover, right? The four humors, blood, bile, was it black bile, green bile, yellow bile? I don't remember. And so, so they called it humoral immunity. So they thought there was this soluble thing. It wasn't cells, because you could see cells under a microscope, but they knew there was something in like the serum of people, hence where like this antibody administration came from, like from horse serum. They knew there was something in the serum that was protecting your body. And so they called that a soluble factor. They didn't know what it was, but they knew it would protect you. And he thought that that was the main way that we prevented disease. So we called it humoral immunity, hence humors. I will often use that term. That's still a term that we use. Antibody immunity would probably be a better way. But I mean, humoral just sounds kind of cool, so we still use it. And he demonstrated that because he's a scientist. He's like, well, if we take the serum, it from this horse and inject it into a person, it prevents diphtheria infection. There's no cells in that serum, so it must be some kind of humoral soluble factor. Well, pretty cool. Well, Machnikov, so he was around too, and he was like, well, I don't know. I just took this starfish out, starfish larvae, which is a eukaryote. It's big enough, and he would inject. He like stuck stuff into the starfish larvae and then looked at it under a microscope. Which, I don't know. He had a lot of free time, and he noticed these cells would come and eat the bad stuff. Okay, and so he was observing phagocytosis by what was probably a macrophage, and so he was like, well, I think that, I mean, if I put this stuff in this foreign object inside this starfish larva, this cell comes and eats it. So it must be cells that are responsible for immunity, or I was going to say immunology. And so he thought it was a cellular immunity. And so they shared, they did a ton of work in all this stuff. And in 1908, they actually shared the Nobel Prize for all of their work. I have a couple pictures here. So this was Erlich postulated side chain theory. So here's Machnikov. He just drew phagocytosis, which is awesome. This is the best picture I could find of his. And then Erlich, he promoted a couple theories. Some of his hypotheses have been disproven, but overall his idea was really, really good in that there is a soluble factor that can prevent disease. And so in the black here is basically what he drew. So this is like the foreign, this would be like a bacteria spitting off antigen. So they look like sperm, but pretend it's antigen. And these black things would be like the antibodies inside your body that are attacking the foreign antigen. And so he drew this in 1900, which was way before we established what any of this stuff was. Now, moving on to the future. So we're moving on, and basically, I found a really nice review article. If you ever want to read it, it's going to take you through the history of immunology. It's really, really nice. It's more detailed than what I have time to go through here. I don't want to bore you too much, but it's called Immunology's Coming of Age. It's Kaufman. He has a joint professorship at like Texas A&M and somewhere in Germany. I can't remember. Maybe it's Pasteur Institute. Anyway, so the big foundational dudes, because I mean, obviously they're all dudes, always, were these dudes. We've talked about a few of these. Erlich was really important. Machnikov and Machnikov, his name is spelled differently in, like he was from Prussia, so Prussia versus French, so that's where his name gets changed a lot. But anyway, so if you want to dig a little bit deeper into each of these people, you can, but you can see here the idea that they came up with. So phagocytosis by Machnikov. Erlich side chain theory, that was basically his idea behind antibodies, though he did not coin that term. But all of these concepts are concepts like serum therapy, complement we're going to talk about, opsonization we're going to talk about, anaphylaxis we may end up talking about. But all of these are really, really important immunology ideas that we're going to be talking about in this course, which is kind of fun. Now, if we keep moving on, so we're going to go past 1900s, we're going to head from 1900 through like the early 40s. So World War II-ish, which kind of, disrupted everything, as we know. I'm calling that mid-immunology history, for lack of a better term. So we knew that antibodies were super cool. And so we decided that we should devote a ton of research to antibodies because they're probably the main defender of the body. But there were a bunch of phenomena that we couldn't explain with antibodies because they were really, really fast. And so we knew that antibodies could just do their thing. They would bind an antigen. That actually became the definition of an antibody, was something that could bind to a specific antigen and actually a specific epitope on an antigen. So an important idea that we're going to be talking about later on as well, but I want to keep hitting this, the idea of what an antigen is and an epitope and an antibody. And so you can see down here, let's say that this is the diphtheria toxin. Okay, and so the diphtheria toxin, it's a big old protein, and proteins can be very, very large. And so they can have different pieces, right? There's different domains on proteins that do different things. Some are structural, some have enzymatic activity, whatever. And so antibodies will bind to very small portions, very like a really tiny chunk of that whole protein. Okay, so if that whole protein, so the diphtheria toxin in this case, that whole protein we call the antigen, but the one little spot that the antibody binds to, we call that an epitope of that antigen. Okay, now if you have polyclonal antiserum, so that would be polyclonal serum. So like if you're taking the diphtheria toxoid and you inject it into a horse and you pull that out and you look at all the antibodies in there, some of the antibodies are against the blocky green thing. Some of the antibodies are against the pyramid looking thing. Some of the antibodies are against the yellow thing. So that's polyclonal. There's more than one clone. We'll talk about why they're saying clones. Has to do with B cells. But don't worry. So we're going to talk about that later. But that's called polyclonal as opposed to monoclonal. Monoclonal antibodies can only bind to one epitope. Okay. Now, it seems like polyclonal would be better. Yes, that's true from the standpoint of maybe trying to prevent a disease. But the development of monoclonal antibodies absolutely changed the face of how we study the immune system and how we study just tons of other stuff. Because if you have monoclonal antibodies, they're very specific against one thing. And so you can kind of take advantage of that to, like if you've ever had a complete blood count, they used monoclonal antibodies to look for very specific markers on your cells, which if it was polyclonal, you wouldn't be able to tell which marker was which. And so they just have different uses. Anyway, so some of the things that we didn't understand was like, why did we reject grafts? So why is there so much host versus graft disease? Why can't I just take a liver out of anybody and stick it into another person, right? They're both humans. Well, that's kind of confusing, and it would take time, and we didn't understand why it took so long. The same thing with tuberculin. Why does it take 3 days for you to get that, you want to get tested for TB, they stick it in your arm, and you have to wait 3 days, right? There's a delay to that. And so that's called delayed hypersensitivity for that very reason. And so that's one of the phenomena that we didn't understand, or several of the phenomena that we didn't understand why that was happening. And so the idea of cellular immunity was becoming more prominent. Now, at the same time that all this was happening, which Landsteiner up here, he's the reason we know like their blood groups, like A, B, O blood groups. He's the one who came up with that, and some of the ideas that led to the development of cellular immunity being super duper important. Now, I'm calling this late immunology. Like this all was kind of, a lot of this happened at the same time. I'm just broadly giving that generalization. But the idea of cellular immunity was the idea that the T helper cells basically are the most important cell to the crux of the immune system. And they're helping not just B cells make antibodies, okay, because those are the kind of cells that make antibodies, but there's also a whole set of cells called T killer cells that will blow up infected cells, usually virally infected cells or cancerous cells. So if your cells are infected because viruses are weird and sort of take over, we have to have a way to kill them. And you can't get antibodies into the cell to actually kill it. So you need a cell to actually come along and blow up the bad guy. Okay? And so that's cellular immunity. Now, the reason a lot of this actually became possible to actually study this stuff was the improvement of technology. And that's something that's going to be a through line through a lot of this, is that without the advances in technology that we had, we couldn't actually study this stuff. I mean, that's evident if you just kind of keep going backwards to all these older people, was the idea that as their technology advanced, they were able to do cool new stuff. And so now we're at the point where these are all just the famous immunologists. I mean, I'm an immunologist and I'm not on this list. There's tons of famous immunologists. There's tons of people. For every one of these people, there have to be at least dozens, if not hundreds of people who helped support their work. And these are just the people that, most of these people are just ones that won Nobel Prizes for immunology. There's tons of other cool stuff. I mean, after we started understanding more about how cells talk to each other and their cellular immunity, we started doing other stuff, which all are topics that we're going to talk about in this class. And so antigen presentation using MHCs, the receptor diversity of your T cell receptors, of your B cell receptors, all the signaling pathways, whether those are macrophages and dendritic cells talking to each other, CD40, CD40 ligand, um, the two, which I would argue is now three signal hypothesis, um, all this interconnectedness between cells of the immune system, which actually is what makes immunology kind of a tricky topic to study. And so there's a lot of moving parts and a lot of interconnectedness. And so that can be a little daunting. And so that's kind of my job, is to try and make it a little less daunting. Now, if we're talking to the present, I mean, modern immunology, that was a question they asked me during my dissertation defense for my PhD was, what do you think is the most important thing in immunology in the last 5 years? And I said plasticity because I thought I was really tired, and that seemed like a really good broad thing to just say. Cell plasticity being you could take like a T helper cell and turn it into like a T helper 22, and then maybe have it go backwards. Um, you see that in stem cells and stem cell work that's been going on, where we could take just a regular cell from somebody and make it kind of go backwards and become a pluripotent, or at least a multipotent stem cell that could then become something else. And so that idea that cells can be reprogrammed because they have all the DNA in there. So how are we controlling what cells are doing? Also, the deeper subset. So as our technology has improved, we're able to take. We used to just have like we knew there were T helper cells. Okay, cool. And then they're like, all right, well, there's T helper ones, T helper twos based on what they're secreting. Okay, cool, or what they have on their surface. Now we know there's subsets of those. Like there's T helper 17 cells, there's T helper 22 cells, there's T helper 9 cells, there's T regulatory cells, there's B1 B cells, B2 B cells, there's a whole macrophage subsets, there's dendritic cell subsets. It's going to drive you absolutely crazy. We're going to talk a little bit about that later. I had to put these in because this was my grad work, was mucosal immunology. Um, I would say the balance between inflammation and immune suppression, that's really important. That would be T helper 17s, like inflammatory versus regulatory suppression. Um, that's a really important field. And the microbiome. Now we understand that like the bacteria that live inside your gut are actually really important for keeping you from getting sick. And so that's kind of cool. And so that's the end of this talk. A lot of history here. I definitely encourage you, if you wanted to, look up this paper, Frontiers in Immunology paper. So they put out review articles. There's a bunch of frontiers. That's a whole journal, a set of journals. Immunology is coming of age if you want a really nice synopsis of the immune system and the history of it.